It belonged to zoologist Thomas Hunt Morgan and his student colleagues. From about 1910 to 1930, they formed a unique and highly productive research team. Through a series of ingenious experiments, they explored and mapped the chromosomes and discovered within them the secret hiding places of Mendel's hereditary factors. They worked in what is now this teaching lab, which back then was a slightly larger room. In this limited space, Morgan and his students advanced genetics from the level of the chromosome to the level of the gene. They did it with the help of a new experimental partner, the tiny fruit fly, or Drosophila. It is far more efficient than Mendel's pea plants. Its reproductive cycle is only 2 weeks as opposed to a year. It breeds prolifically, and huge numbers can be grown in a small container. As well, its chromosomes are particularly easy to study under the microscope. But unlike pea plants, fruit flies didn't come in different varieties, so Morgan and his students set out to produce their own, without much success. Then a breakthrough finally occurred. They noticed a male fly with white eyes instead of the usual red. When they crossbred this mutant male with a normal red-eyed female, all of their offspring, the F1 generation, had red eyes. According to Mendel's theory, this meant red was the dominant trait and white the recessive. But when the offspring mated among themselves, the next, or F2 generation, showed a pattern that Mendel would not have recognized. About half the males, but none of the females, had white eyes. According to Mendel, gender should not have mattered, and in this generation about a quarter of both the males and females should have white eyes. But that is not what Morgan and his students found. However, they were able to explain this exception to Mendel's principles by adding in the newly discovered sex chromosomes. They concluded that the white eyes in half their male fruit flies was indeed a recessive trait as Mendel understood it, but one that is located on an X or female chromosome.